pairing of sulfhydryl groups to form disulfides is not an invariant property of the linear chain, but depends on extrinsic factors including the redox. In other words, protein synthesis and specificity in general, and antibody synthesis and specificity in particular is redox dependent. If this is so, then any agents who will induce the same redox changes as a virus could induce the synthesis of viral antibodies and antigens in the absence of the virus. Viruses including RNA tumor viruses share antigenic determinants with normal host cell components, a phenomenon known as molecular mimicry. The same phenomenon may exist in the case of the HTLV3/Lab virus. The most prominent and persistently detected antigen in AIDS tests is a protein of a molecular weight of 41.000 P41, which is approximately the molecular weight of polymerized actin, a protein found in all cells including bacteria. A protein of the same molecular weight, isolated from a number of viruses, has been shown to be actin, and to a major constituent of many viruses including RNA tumor viruses. It is of interest to note that the polymerized form of actin increases with oxidation. Of interest is also the fact that metagenic stimulation of normal cells with clone leads to the expression of uncoverable antigens without virus particle synthesis. The presence of natural antibodies in the sera of physiologically healthy animals, directed against a variety of antigens, has been well established and documented. Antibodies against the uncoverable proteins are widespread in non-infected human sera and vary with age. Furthermore, substances as diverse as normal components of the serum, extracts of bacteria and non-protein molecules such as glycogen are important factors in determining whether a given human serum registers positive for oncovirus infection. Snyder et al. Discussing their work on human uncoverable antibodies concludes, the results are consistent with the idea that the antibodies in question are elicited as a result of exposure to many natural substances possessing widely cross-reacting antigens, and are not a result of widespread infection of man with replication-competent oncoviruses. Barbacid et al. State. This finding not only demonstrates that the antibodies were directed against cellular, rather than the virus-coded antigenic determinants, but also exclude the possibility that this immune response was elicited as a consequence of oncovirus exposure. There are two blood tests routinely used for AIDS detection, ELISA and Western blot, neither of which detects the virus itself. Although the latter test is more accurate, both give persistent false positive results. The false positive problem has led to harrowing decisions about what to tell patients whose samples appear positive, although manufacturers stress that the current tests are not intended for use in diagnosis. It is significant that the false positive results increase with age and stickiness of the serum, and the stickiness's viscosity is redox dependent and increases with oxidation. The outcome of the test seems also to depend on who is performing them. Thus, one group found 7/10 sera positive for viral antibodies, whilst another group testing the same sera found none. Most importantly, Bigger et al. Found that the probability of having a positive ELISA for HTLVI, HTLV2 and HTLV3/Lab increases with age, poverty, immune complexes concentration, and especially with malaria and other parasitic diseases. They conclude, if the human retrovirus reactivity observed in illicit tests is frequently nonspecific among Africans, the causes of the nonspecificity need to be clarified, in order to determine how they might affect the seropidemiology of retroviruses in areas other than Africa. The only sensible conclusion is therefore that seropositivity does not mean virus positivity. However, Gallo and his collaborators are of a different opinion and state, we should proceed with blood bank antibody tests. They base their opinion on the fact that HTLV3/Lab can be isolated from the peripheral blood of greater than 80% of people with serum antibodies to the virus. Although this is true, it is important to note that all the isolations are done in vitro, see below, after some unusual and drastic manipulation of the lymphocytes obtained from the patients. The initial reaction to the retrovirus hypothesis was one of skepticism. However, after the publications of the 1984 papers, Science for Me, the theory became almost universally accepted. In these papers, in vitro experimental evidence for the detection and isolation of HTLV3/lab is documented. But in a paper subsequently published in the same journal in the same year, Science 7 December, the Americans, by using the Southern blot hybridization technique which can detect as little as one copy of viral DNA per cell, obtained negative results on fresh peripheral lymphocytes, lymph nodes, Kansas, bone marrow and spleen from AIDS patients and AIDS-related complex ARC. They conclude, thus the lymph node enlargement commonly found in ARC and AIDS patients cannot be due directly to the proliferation of HTLV3 infected cells as occurs with HTLVI in adult T-cell leukemia. Whether the lymphocyte proliferation in lymph nodes occurs in response to infection with HTLV3 or another agent, or both, is not known. Similarly, the absence of detectable HTLV3 sequences in Kaposi's sarcoma tissue of AIDS patients suggests that this tumor is not directly induced by infection of each tumor cell with HTLV3. Furthermore, the observation that HTLV3 sequences are found rarely, if at all, in peripheral blood mononuclear cells, bone marrow and spleen provides the first direct evidence that these tissues are not heavily or widely infected with HTLV3 in either AIDS or ARC. In an article published this year by the French group, it is stated, "It is unlikely, however, that AIDS is the result of a direct progressive destruction of T4 cells by the virus for at least two reasons." Thus the originators of the viral theory of AIDS agree that there is no direct evidence to support their theory. What then about the claims of repeated isolation of HTLV from AIDS patients? All the experiments for detection, characterization, continuous production and isolation of HTLV3/lab are done on in vitro cultures. Furthermore, the cultures are not solely with T cells from AIDS patients, but cultures with highly selected neoplastic T cell lines. It must be emphasized that unlike other viruses, HTLV3/lab has never been isolated as an independent stable particle. By isolation of the virus, in fact, it is meant transient detection in the cell culture of viral antigens, viral antibodies, the enzyme reverse transcriptase (RT), and of virus-like particles budding from the cellular membrane into the extracellular space. In the vast majority of cases, isolation is synonymous with RT detection. However, apart from RT, these cultures have almost any other enzyme implicated in DNA synthesis, and it has not been excluded that viral reverse transcriptases are cellular enzymes. The viral specificity of RT is believed to be given by the template primer it uses. For HTLV3/lab isolation, the French and the Americans use either DT right (12 to 18. A right (and or DT right (15 A right (n) as template primer, but in earlier papers Gallo and his collaborators present evidence that DNA polymerase gamma prefers exactly the same template as the one used for HTLV3/lab isolation. It is also significant that the kind of template a polymerase uses and its activity depends on the culture conditions, and probably on the state of cellular development, i.e. the activity of the enzyme depends on the normality of abnormality of the cells. In rare cases, by isolation is meant finding a virus-like particles either in T cells in vitro, or cells other than T cells in fresh AIDS tissue. These particles are not only hard